All right, it's my pleasure to introduce Emily Churchman. She's a PhD candidate at Auburn University, co-advised by Dr. Mark Lyles and Dr. Miles Lang. Her research interests broadly include host pathogen interactions and vaccine development in aquatic animal health. She is currently a NIFA predoctoral fellow, the first one at Auburn, and will be discussing her work on recombinant protein vaccines against Collimaris disease. Thanks a lot for being here, Emily.
All right. Thank you, Nick, for that introduction. Thank you all for being here today. I'm very excited to share this story and continue the work that Miles has previously started in the lab. S- So Miles gave a great introduction for aquaculture, so he made my job a lot easier. So I'm just gonna briefly touch about Collimaris disease and in relation to the catfish industry. So both the Kelly lab and the Friends labs both did separate surveys across about 5 to 7 years, I believe, looking at Alabama or Mississippi and Alabama, or Alabama, Mississippi, respectfully, looking at disease cases and total losses in catfish for Collimaris disease. So at the top figure here, we can see we have increasing total number of fish loss for in West Alabama. And down here, Dr. LaFrance also was seeing which of the CCBs were most prevalent in disease outbreaks, Collimaris disease outbreak in channel catfish, and found F. covey is by far the most prevalent and the most virulent in channel catfish. So very briefly, just a couple of clinical signs we see. We see a lot of uh, biofilms and lesions on gills and other mucosal surfaces, fin rot, cigar mouth. You can see sometimes you get these lesions around the dorsal fin, look like saddleback. And interestingly, you take samples of a lesion for a gill, you can get these beautiful haystack columns, which is indicative of CCBs. And like I said before, we're just focusing today on Phylobacterium covey. Now, Miles already discussed this, but there were live attenuated vaccine candidates against Collimaris disease, but they have had mixed efficacy due to the genetic diversity of our CCBs. And again, as Miles said, he's been working with F. covey chaperone protein DNA-K, has shown a protection in a laboratory challenge and has been measured by a B cell response. But this has not been tested as an orally delivered vaccine. So, a important hallmark for F. covey infection is the ability to form a biofilm on the gills. And the biofilms are great at protection from environmental stressors, UV, pH changes, and microbials, and also, more important in our case, the host immune response. We see that VAS that show are more susceptible to F. covey infections tend to have those worst clinical signs of biofilms of the gills. And previously, Dr. Lang has done a study, a transcriptomic analysis of F. collimaris and F. covey isolate, actually, the biofilm state when stimulated with catfish mucus. So we have a whole list of Gs that we think are important in virulence because they're in the biofilm state. Thus, we think they could be great potential vaccine candidates. So we have selected two F. covey antigens upregulated in the presence of catfish mucus to test as potential recombinant protein vaccines. All right, I have to give a disclaimer here. I wish I could tell you the name of the proteins. We have a patent pending for the USDA. I do not own it. The government owns it. But briefly, how we express them, we use a sumo tag gene cloning system, which is a solubility tag. So we clone them into our expression vectors. And similar to using like an E. coli machinery, we're able to purify using that 6 his tag. And then we, in theory, not theory, we could cleave this using the kit protease and just have our protein of interest. And these were for an IP injection study just to test the endogenicity of these two proteins. But if due to time constraints. I won't explain fully, but we decided to move forward towards using the PET28 vector for other trials because we were actually having an incomplete proteolytic activity with our sumo protease. Thus, we were getting a lot of anti-sumo antibodies. So just for this next slide, just that has the sumo tag, but future studies, we did not use the sumo expression vector. All right, so for immunogenicity of our first protein, so we vaccinated 30 fish with our either an adjuvant only control or our commercial adjuvant with the FCP1. So, and at 30 days post-immunization, we sacrificed collected sera and ran ELISAs and Western blots. So, and this is, mind you, these are only anti-FCP1 antibodies. These do not include the anti-sumo antibodies. So as we can see, our fish were able to produce antibodies specific to our protein, our antigen at 30 days post-immunization here compared to our control. And when we ran catfish sera and Western blot when we ran our protein, for the Western blot with our catfish sera, we were able to get our expected band size of about 70-ish kilodalton. And similar for our second protein, which actually had a smaller band size, about 25, 30 KD. And we're also to get an antibiotic response. So that's great. We know our fish can make antibodies, but for the industry, injection is not practical for channel catfish. So we decided to move forward with our bath immersion trial. So this picture is a little dated. This was actually before we had our house remodel, but same idea with our RAS system and individual troughs. So we had one control with our IMS adjuvant only, and then we had 3 vaccine groups that received either antigen or a combination of both antigens at 1 microgram per mil for 30 minutes before being stocked in a flow-through system for a couple days and then moved to recirc water. We had several sampling periods throughout the entire study. We collected sera and skin for antibody titers from 15 fish, and then from 4 to 5 separate fish, we collected a separate skin sample for qPCR of immune-related genes. And then to test the efficacy of our vaccine, we did a laboratory challenge at 9 weeks post-immunization with a industry-relevant isolate. So for our challenge, we hit the gold standard. It works. So we were able to have increased survival during our laboratory challenge compared to our adjuvant-only control for all vaccine groups. Interestingly enough, though still protective, our combo group with both vaccines, or both antigens, excuse me, was less protective than our individual counterparts, which to me is fascinating. We still don't necessarily know why. We have a hypothesis there's some sort of protein-protein interactions going on, but that is stay tuned next year. We might have better answers for that. Okay, next we want to take a look at our qPCR data. So these are right now I'm going to show you some innate immune genes. So these are toll-like receptors, which recognize different pathogen-associated molecular patterns, or PAMPs. And just to orient everybody, this is our increased fold change compared to our adjuvant-only control, which has a baseline of 1, and either you'll see an increase expression or level or decrease. So as we can see, immediately after vaccination, we do see an increased expression of our toll-like receptor 1, and the other 3 we also looked at. But then it kind of leveled off, maybe peaked around again at 9 weeks post-vaccination. But there wasn't necessarily a clear trend. And when we looked at other innate immune genes for a pro-inflammatory response, we're kind of seeing the same thing, but we're still currently in the process of teasing out what is going on in the host immune system. Now, for B cells, so we did several B cell markers for like IgM, MHC class 2, and same idea. We're not entirely sure. Specifically for IgM, we see a huge increase, is fold change for, and that should be FCP2 or 1 and 2, sorry, typo. And then levels off, and then and then some increase at 9 weeks again. But there's no consistent trend between vaccine groups. So we are currently still in the process of figuring that out. And interestingly, too, we weren't for the skin and the sera, and I'm just going to the skin ELISAs right here on the right, we were not able to get any detectable mucosal antibody response or sera antibody response throughout the duration of the study. Now, that could be we are below the detection of a microtiter ELISA that could, which we are looking for alternative means for measuring mucosal immunity. And I didn't show this today due to time, but we're also looking at different T cell markers and expression and seeing maybe this could possibly be a C or cell mediated immunity instead of our B cell mediated immunity like DNAK was. So overall, this study has led to a lot of interesting directions. So I just want to highlight my 2 undergrads. I like to brag on them. They're awesome. So this has actually led to 2 different projects for them individually. Right here, Ashley is going to be looking at those protein-protein interactions from the multivalent vaccine of FCP1 and FCP2. And she's actually received an undergrad research fellowship from Auburn for the study. So go, Ashley. And then Victoria, she's also awesome. She is, when I was mentioning about other means for detecting mucosal immunity, we actually have an octet, and she's going to be trying to develop a protocol for using biolayer interferometry to increase our sensitivity of antibody measurement and look at different kinetics and so forth. Now, what my focus is going to be on for this fall is actually doing a dose-dependent response for an orally delivered vaccine. Immersion's great. It's the industry standard. We have shown it has worked and it's protective. But with an orally delivered vaccine, you're essentially just eliminating any extra labor a farmer has to do. So that is something we are very excited for, and we'll be starting this fall. All right, so just a bunch of people to thank for. Thank you for, I want to thank the Lyles Lab and all the scientists at USDA. I want to specifically highlight Nathan, who you'll hear later today, but he did all that qPCR work. He's awesome. And with that, I will take any questions you may have.
Excellent presentation, Emily. We've
Thank
got
you.
time for one quick question. If someone would like to unmute themselves or drop it in the chat.
I have a quick one, Emily.
All right.
War Eagle.
War Eagle.
Yes. Excellent talk. I really like it. I have a quick question for you. So usually for columnaries, we used to read in the literature that antibody titers does not correlate with protection. So in your hands, from the experiments you've done, what did you see? Did you see a correlation when you get an antibody response with protection in fish? And is it consistent?
Yeah, so that's kind of where I was talking about earlier. We weren't able to get a measurable antibiotic response. So that's where in the past with DNAK, there was an antibody response and there was protection against columnares. With these 2 proteins, we're not seeing that, which is why we're looking more towards maybe this is a T cell mediated immunity. So it wouldn't be antibodies, it would be T cells.
Sweet. Thank you so much.
You're welcome. Also, for the record, I'm actually not in fisheries, so don't have to worry about the conflict for the sponsor.
Great. All right, well, thanks again, Emily. Great presentation. Thanks for doing it.